Okay, so let's do this. Let me make sure you know recording. Okay, so let's go over the quiz so that you can see that the quiz, you can see how the questions are. Okay. So let's go over the quiz so that you can see that how the questions are phrased. You can see that the questions rely a lot on understanding more than memorization. And you can learn in the future how questions like this might come up. So, in the first question right here, let's see, RFLP was performed on family to understand the association of a mutation with a disease, right here, okay? There are two possible alleles, A and B. So here we go, sudden blotting was performed. Okay, let's see. So you have allele A, where you have this chromosome right here. If cut by a restriction in the nuclease at this position and that position, you would have the generation of a 5 kilobase DNA fragment that can be detected by a probe right here. Okay. Now allele B, you can have you have 3 restriction sites, 1, 2, 3. So the probe would detect a 2 kilobase DNA fragment. Good. This is the location where the probe binds to, okay, right here in black box. That's a pedigree. This is the family. You already know what a pedigree is. So right here, that's the male, that's female. If it is shaded, it means the person is affected. So affected male, healthy female, healthy male, affected female, and so on. And these are the numbers of individuals. Okay, and that's the pattern. So this person right here is heterozygous. Why? Because this person has a large, the allele A, 5 kilobase, and allele B, the 2 kilobase. Right here, look at this female, homozygous, having 2 allele A chromosomes. This individual right here is homozygous as well, having allele B. Okay, so let's see. Individual 2 is homozygous for the mutated gene. Where is it? That's individual 2, right here. So, having only one fragment, 5 kilobase, so actually, she is homozygous for allele A, not allele B. So, that's the mutated allele right here. To have a disease, an individual must carry 2 copies of allele B. Well, that's not right. Why? Look here. This male is affected, but this person is heterozygous, having only 1 copy of allele B. So that's not right. Okay, same thing here. 2 copies. Now, this female is homozygous, but she has 2 copies of allele B. So that's not right. It is expected that individual 8 is heterozygous. Well, let's see. Individual 8, healthy. So what do you expect? The person must be homozygous for allele A. Okay? So, is she heterozygous? No, she's not. She's homozygous. If individual 3 is married to a normal female, all children will be affected. Well, let's see. This person right here, healthy. Homozygous for allele A. So a normal female would also be homozygous for allele A. So there is no way that children would carry allele B. Okay, because they don't have it. If a probe covering the region marked XXXX is used, you can reach the same conclusion. Well, let's see. Right here, if the probe is used covering this region right here, binding to this region, you can reach the same conclusion. Yes, because instead of having a 2 kilobase DNA fragment, you will have a 3 kilobase DNA fragment, and you can still differentiate it from allele B. So yes, you can reach the same conclusion, and this is the right statement. So that's what we're looking for. One of the following is true. Okay, the sequence this exists in a human genome. One of the following is true. So we're looking at the true statement. The opposite strand, that is the complementary strand, is this. Well, notice something in here. Again, this is a trick that I told you about. I can use it, and I told you I can be mean. So I'm not indicating where the 5' and 3' ends are. So immediately you know that this is the 5' end, this is the 3' end. Look at the sequence. Again, there is no indication for 5' and 3'. So this is the 5' end, this is the 3' end. In order for this to be complementary, it has to be flipped. It has to be 3' here and 5' here. Okay? Otherwise, they're not complementary. So it's made of ribonucleotides. That's not right, because that's human genome. It must be made of deoxyribonucleotides. It exists in telomeres. No, it's not the repeat of telomeres. It is part of a minor groove. I have no idea if it is part of a minor groove or not. So all of these options are wrong. That's the right one. The last T in the sequence, that is this one right here, this T, exists in a monophosphate form, and that is right. Because if you look at the structure of DNA, you would notice that the first nucleotide is actually, the last nucleotide, every nucleotide is actually a monophosphate, not a diphosphate, not a triphosphate. So look back at your slides. A nucleosome contains all of the following except, I'm going to shoot myself if you didn't know. Well, I would have shot myself if you didn't know the answer. You killed me with questions about nucleosomes and chromatosomes, H1 and linker DNA, and the differences between them. Well, the difference between them, one of the differences between them is that chromatosomes have H1, nucleosomes do not. Okay? A nucleosome does have a DNA free of histones. That's right, that's the linker DNA. A DNA is complex, it's a DNA complex with histones. Well, that's right, because you do have a histone octamer. A histone octamer, it's right here. Okay, H2A, H2B, well, yes, it's part of the histone octamer. So, that's the right answer. A non-coding RNA is one of the following. The definition of a non-coding RNA is that it does not produce proteins. It's not expressed? No, because it can be transcribed, so it can be expressed, okay? Except that it cannot be translated. It doesn't have a specific gene? No, it does. It comes from a specific part of the genome. It can be transcribed. It is produced from another RNA. That's not right. It's produced from DNA. It has a repetitive sequence. No, who said that? No one said that. So the definition of non-coding RNA is it does not produce proteins. Simple definition. So a DNA sample has a concentration of 250 micrograms per ml. It was diluted 1:50. What do you expect the absorbance of the diluted sample to be at 260 nanometer of light? So this is a what we're looking for is not the concentration. We already have the concentration. What we're looking for is the absorbance. So what's the concentration of the diluted sample? It is 250 it's 250 microgram per ml. You diluted 1:50. So what is now the concentration of the diluted sample? It's 5 micrograms per ml. Good. Now we already know that one unit of absorbance is equal to 50 micrograms. So the concentration is 5 micrograms per ml. So basically the absorbance would be 0.1. So it's tenth the absorbance of the 50 micrograms per ml. So let's go over this one right here. So we have three denaturation curves of 3 DNA fragments, X, Y, and Z. This is the melting point right here for all three of them. That's the temperature. So it is 70 for the first one. It's about 85, 87 for the second. And for the third, it's about 110. Okay, so let's see. So we're looking for a statement that is true or it can be true. Okay. The increase in absorbance at higher temperature is because DNA becomes single-stranded. Yes, definitely. I mean, this is the whole concept. Why is it that you have more absorbance? Because DNA becomes single-stranded. So this is a true statement. What else do we have? Sample Y has higher CT content than sample Z. Well, it's the GC content, not CT content. The melting temperature is approximately 120. No, it's 120 is when they're all fully denatured. The melting temperature is the 50% mark. At 80, the majority of DNA in samples X and Y are denatured. That's not right. No, it's not right. Why? Well, at 80, right here, the majority of the DNA in sample Y is still not single-stranded. Not fully single-stranded. Here, at 50% absorbance, half of DNA molecules are fully denatured and half are fully double-stranded. Well, no, we're not talking about fully denatured. Part of the DNA fragment is denatured and part is not denatured. Okay? So, shown is a 4 kilobase DNA fragment completely digested by two endonucleases, BAMH1 and HindI3, added together. So they all cut the DNA. The fragments generated have the following sizes. So this one here would cut the DNA right here at this position, generating a DNA of 1.8 kilobase. Okay. Now, this same DNA fragment can be cut by HindI3, generating a fragment that is about 1 kilobase. Okay, this enzyme would cut right here, generating a DNA fragment that is about 0.3. And then you have the remaining DNA fragment has a size of 0.9. So the right answer is this one right here. Okay. Number 11. A certain nucleotide outside the regulatory or coding region of gene X was analyzed in 1,000 healthy individuals. These are the controls. And 100 patients. These are the cases. G was found in 94%. And A was found in 6% of the healthy individuals, of the DNA in healthy individuals, okay? And in cancer patients, it's 61 and 39. So, let's see here. So, we're looking at what? We're looking at a single nucleotide polymorphism in here, okay? So, we're looking at a certain nucleotide, SNP, single nucleotide polymorphism. The genetic position is a single nucleotide polymorphism? Yes. Okay. Why? We're looking at a single nucleotide and we're looking at something that exists in at least 1% of the population. So healthy individuals, 6%. That's more than 1%. This is a genetic variation in gene X among the population. Yes, that's right. It is. Gene X is involved in the pathogenesis. Well, yes, because you look at the numbers in here. A exists more in cancer patients, a lot in cancer patients versus healthy controls. So it seems to be associated. Okay. Is it involved? Is it involved in pathogenesis? Well, we don't know that. It is associated, but we don't know if it is pathogenic or not. Why? Because look here, outside the regulatory or coding region of gene X. So it is linked. It is not causative because it's outside the gene or the regulatory region. So can we use it to predict the susceptibility? Yes, we can. So this is a link. Well, that's right. It is a link, but we don't know if it causes cancer or not. Okay. Actually, it may not because it's outside, again, the regulatory region. Which of the following is not correct about intergenic DNA? The right answer is, whoops, okay, let's see. The right answer is, so the majority of the human genome is composed of intergenic DNA. Well, that's right. What is intergenic? That is outside genes, between genes. Intergenic DNA does not code for proteins. Well, that's right. It's given, okay. The repetitive DNA in the intergenic DNA can be dispersed or tandemly repetitive. Well, that's right. Yes. like transposons, for example, like telomeres. They are repetitive and they are intergenic. Centromere is a type of tandem repeats that is identical among different individuals, among different chromosomes, I'm sorry. No, it is not identical. It is not. It is similar, it's different actually among chromosomes. And that's why we can label chromosomes specifically using probes for their centromeres. Mini satellites and microsatellites are variable among individuals. But yes, that's right. That's a true statement. So this is the wrong statement right here. What else do we have? Let's see. So transposons, most of them can move. That's not right. We're looking for the true statement. They have to be inserted to be in the coding region of genes to be pathogenic. Well, okay. Well, no, that's not right. Why? Because they can be inserted in the regulatory region. Okay. They are examples of interspersed repetitive DNA sequences. Well, that's right. They are repetitive and they are interspersed. The majority of them originate from DNA viruses. No, they originate from RNA viruses, retrotransposons. They are 20 base pair long regions. No, they're not. And there are two types. We have signs and we have lines, right? So short, but the short can be about 300 base pair, like the allele sequence, or large or long interspersed regions, okay, or elements, and they are larger than 300 base pair. So no, they're not 20 base pair long regions. STRs and PNTRs are used for genetic markers. Why? Well, because their sizes are unique among individuals. That's right, their sizes, they can be the number of repeats would be different among individuals. Probes are easy to make for them? Well, no. I mean, that's not the reason why they are useful biomarkers. They are located within or nearby active genes. Not necessarily. Their sequences are variable? No. We said that their sequences are the same, but the number of repeats is different. They are gene related? No, they're not. They have nothing to do with genes. Complementarity is a feature of DNA that indicates the following. DNA is anti-parallel? No, it has nothing to do with it. DNA is helical? No, that's structural. Number of A and G would be equal to number of T and C? Well, that's right. I mean, it is an indication, because if you have the numbers of A would be equal to the number of T, number of G would be equal to the number of C, so this complementarity suggests this. A minor groove is opposite to a major groove. It has nothing to do with it. Bases are almost perpendicular. This is again structural. It has nothing to do with being DNA being complementary. Which of the following double-stranded DNA sequences needs to be at higher temperature to separate into single-stranded DNA? Well, it's either it depends on number of factors: ionic strength of the solution, the length of the DNA fragment, the GC content, pH. We talked about this. So you look here, the only variable is what? The length is the same. The only variable is the GC content. So the DNA fragment with the highest number of GCs would need higher temperature to be denatured. A human genome is cut by resection in the nucleus and then analyzed by southern blotting. You can know the following from southern blotting. You can know the sizes and numbers, but not the sequence. I wouldn't know the sequence of the whole DNA fragment. I can probably tell what the sequence of the probe binding site, but not the whole DNA fragment. I would know the size and number of DNA fragments that are generated, but not the whole sequence. And even the whole sequence, I mean, if it is even imperfect hybridization, it can be different a little than the probe. Okay, so that's the Okay, so this question right here, a child wants to know his real father. So that's the genetic profile. Again, we need to match things. Here we have these DNA fragments in the child. This DNA fragment matches this from the mother. This DNA fragment matches this and that individuals 1 and 2, men 1 and 2. It doesn't exist in individual 3, so X number 3. This person is out. Look at this fragment. It comes from the mother. Great. Okay. Look at this fragment. It comes from individual 1, but not 2. So X on number 2. DNA fragment comes from number 1, right? Now you have this DNA fragment. It doesn't exist in any one of these individuals, so it could be a mutation. So you can ignore this DNA fragment. What else? Look at this DNA fragment. It exists in individual 1, but not 2. It exists in number 3. So the best match really is individual 1. So that's the quiz.